Welcome everybody to our second webinar coming to you, well, I'm coming to you from New York City. I don't know where everybody else is scattered about the world. I'm Dr. Oliver Rebooting at the Cooper Union in the Kambar Center for Biomedical Research, where we also do our journal clubs. And this webinar is brought to you by Life Extension Advocacy Foundation. And joining us today are Dr. Michael Lascartes, Dr. Amy Purl, and Dr. Cosmo Mielke, who are all experts in the microbiome, or basically things to do with the microbiome. And the topic, of course, today is the microbiome. And I guess most of you already know my background. Those of you who don't, you can obviously pull it up at the lifespan.io website. But I think maybe we should do a little round the table intro, just a quick intro, you know, starting with Cosmo, who's in my toolbar up there, is on the left, and to the right of me, and maybe a quick intro, and then we'll go on to Dr. Amy Proal and Dr. Michael Lascartes, and just a quick intro as to who you are and what brought you here. So, Dr. Mielke.
Hi there. Yeah, so I used to study astrophysics, but then switched into biology because I was totally fat and tired of it. So did my PhD in diabetes and obesity and focused on genetics there. And then after that, founded a nonprofit to study it further. And that sort of faded into aging research because I found out that everything is beautifully connected in that regard. So I tend to focus on genomics because it's truly the only causality that's provable in biomedical data sets, because nothing is ever going to reach back and change the genome in a predictable way. It's always chronological with time in terms of causality. So I like to study all the GWAS studies out there and figure out what genes are associated with each disease. And then I hit the molecular biology literature as deeply as possible to try to figure out what those actual mechanistics of the disease are. And that sort of led me into the microbiome and virology side of aging. And I think there's some really strong overlaps there, and profound evidence, in fact, that senescence itself is actually a viral infection.
Very intriguing. I think we'll try to unpack that a little bit later. Dr. Amy Purl, you're up next.
Hi, I'm Amy Proal. I'm a microbiologist. I started studying the microbiome back almost in 2005 when it was barely on anyone's radar. Also, viruses at that time in the human body. Part of why I became interested in it is I was interested in an illness that's not well understood called MECFS at the moment. People get very sick, and it's almost like they have aged prematurely. There are patients who are, their grandparents function better than them. And so I began to look at the role of infection in basically inflammatory disease, and also in neuroinflammatory disease. And of course, that just segued me more into the microbiome, because pathogens persist often in the microbiome environment communities in our body. And I'm interested in how those pathogens act and the way that they can basically, in simple terms, hack into our, affect our gene expression in ways that can drive inflammation, disease, inflammaging. And basically, because I'm interested in diseases of the aging, it just segues me into aging. And I'm interested, there's a lot of overlap, I think, between those conditions and what we're seeing.
Very cool. All right. So 2 intros and already 2 cool topics, MECF and viruses and the potential link to senescence, that I find intriguing because I don't know much about. So I definitely want to know more about it a little bit later. Dr. Michael Lascartes.
Yes. So my roots in this did not start in the microbiome. They kind of evolved. So as a grad student about 10 years ago, I was interested in studying the oxidative stress theory of aging, which I did. But then after I got my PhD, I wanted to study use a less reductionist approach, because we were using gene knockouts, knocking out one gene to study oxidative stress. So I decided to go into the metabolome, kind of for the opposite reasons that Cosmo was talking about, just because that's the actual process of what's happening or what happened, as opposed to what can happen, even though I have obviously respect for genomics. So as a postdoc, I started studying the serum metabolome as it relates to muscle mass and function in aging in people. And as I started publishing papers in both young and old subjects, I started seeing a role for gut bacteria-related metabolites. So with that in mind, I wrote a couple grants and got them funded to actually study the microbiome in aging and in terms of muscle mass and function. And I've got some of that data in review right now in submission. So that should be interesting. But I'm not just interested in gut bacteria, as these guys have talked about, but that's only part of the story. And as I started to dive deeper into how the gut microbiome may affect muscle mass and function, I started reading more about viruses and fungi and seeing that their presence was up in the blood of older adults. And actually, I wrote a book so that kind of links all the information as best as possible to suggest that many of the diseases of aging, including cardiovascular disease, cancer, Alzheimer's disease, et cetera, stroke, are related to alterations in the microbiome, not just the gut microbiome, skin, mouth, gut, lung, all of that. So, but I should say Amy's been on this case for way longer than me. I mean, man, she's been talking about it for, probably more than a decade. So, much respect there too. So I'm new to the game.
All right. Very good. Excellent. That's a lot of great introductions and a lot of great topics that we can spend 2 hours just unpacking those topics. Before we deep dive into that, we have an audience out there who might be new to the microbiome. There's a lot that, I don't understand much about. And I think Elena put a link to a book here, and I think that's your book in the chat. So if anybody wants to check out Mike's book, there it is. Okay, so we're going to start out with introduction. First of all, what exactly is the microbiome? We've heard, on various pop science, websites that the microbiome is, 10 times more cells, that are bacterial in origin, prokaryotic, although certainly there's other organisms as well that are involved in the microbiome, are part of the human, part of your body, as opposed to your cells, which is about 50 trillion of your eukaryotic cells versus 10 times as many microbes that are in and about you. But, other than that, what exactly is the microbiome?
So just to take a step back from that, the microbiome doesn't just involve what's in and on us in terms of bacteria, viruses, fungi, etc. But we live in a basically a microbial world. So the microbiome is just essentially, as you mentioned, the little buggers that are in or on us. But there are something like 10 to the 31st bacteria, viruses, fungi, and others that are on Earth. When you consider there are 7 times 10 to the 9 people, we're outnumbered by a magnitude of 22-fold, or no more than 22-fold, 10 to the 22nd power. So we live in their world. That we've even evolved to exist is amazing to me. So yeah, the microbiome, now scaling it back, is indeed the things that the little buggers that live in and on and inside of us. And for the most part, it's mostly symbiotic. But in the cases of disease or aging, the composition and functions of what lives in and inside of us gets out of whack and becomes a self-perpetuating loop to further decrease function and health and all that stuff.
I'll jump in. So the microbiome is, again, the organisms that live in and on us. And to clarify what those are, we have bacteria, but we also have viruses. Then we also have what are called bacteriophages, which are viruses that infect bacteria and modulate their activity. Then we have fungi. Then we also have archaea, which are sort of like bacteria, but sort of different. And then there's even now evidence showing that there are mycoviruses, which are viruses that infect fungi. So you can there are giant viruses actually just shown in the gut. So there's all kinds of organisms that live in these communities. And microbiome research started in the gut, which is logical. And also the mouth was an early site. People looked at that mostly because we accepted for a long time that organisms live in our gut, in the mouth. So research began there. But as we continue to basically use computer-based tools to look for microbes in the human body, we just, you can't keep up. There are so many organisms still identified almost on a regular basis, to the point where bacteriophages, for example, those viruses that infect bacteria, the current estimate is there are trillions of them in human tissue and blood every day. And also the microbiome has extended beyond the gut. As Mike mentioned, there's a lung microbiome, there's a bladder microbiome that can be connected to UTIs. There's that seems to be microbes in a lot of body sites. A little bit of a debate over the placental microbiome, but strong evidence that microbes also persist in the womb, which can affect infants. And there's also, even at Harvard University right now, a brain microbiome project being done where they are actually finding, they have some pretty, actually I've seen compelling early data showing that there is a microbiome also in the brain. So basically we're filled with these organisms and they interact continually with our pathways, and as much as they're framed sometimes as being friendly, a lot of these organisms are what are called pathobionts. They can change their gene expression to act differently under different conditions. And so the way we live and different things, the medications we take, what we eat, all those kinds of things influence how our immune system can manage these organisms, and they can definitely change their gene expression and act in ways that favor their survival over ours. And that is where we see changes in these communities that can drive diseases and even and aging.
Just to add a little more, not only can they do all those things that you just mentioned, but they can go stealth, where they can't be detected unless we're using genomic-based tools or RNA-based tools to identify their presence. So they go stealth and they lay dormant for sometimes decades, and then they can reactivate, in conditions of immunosuppression for others.
So when you say stealth, do you mean they get into a spore-like state? Do they?
They don't grow. They don't differentiate. They don't. They probably make products, microRNAs and other products that circulate to cells to basically sense the environment. There's some data on that, but yeah, you could take a blood culture and nothing will grow, but yet you do the DNA or RNA-based analysis and there's a bunch of them there. So.
Right. That reminds me of the analyses that are done in the microbiome out in the world, out in soil samples, right, where supposedly you can't cultivate 99% of what you can sequence that's found in the soil sample. So the conditions just are not optimal. We haven't optimized them for growth.
Right.
So we hear a lot about microbiome and how it can basically lead to diseases, how it basically is beneficial. So, there's a yin and a yang happening here. So I've got a list of questions here, but these all kind of go together. And one of the questions I have on my list is what sorts of diseases are linked to changes in the microbiome. But kind of going along with that is, what are some of the benefits of the microbiome? So I guess maybe kind of start off however you want to. If you want to start off with the good versus the bad and just give kind of an overview of that for our listeners, I think that would be another great jumping off point before you start getting into the aging aspects of the microbiome.
Well, the way I look at it, you'll see the microbiome often sort of described as friendly, and that is in a way a bit inaccurate. What it really is that these organisms have evolved, as Mike mentioned, to live with us since basically the beginning of time. And so our pathways have become integrated. So they produce a lot of metabolites, proteins, different amino acids, different byproducts of their metabolism have become part of our human signaling pathways as well. So we just evolved to depend on them in a lot of cases for to break down certain compounds, to produce different neurotransmitters. They do all these kinds of things in our bodies. They're not doing it to be friendly. They're doing it because that's what they do to survive. If the body's in a state of balance, think of someone like an Olympian who's functioning well. The microbes that are in their bodies are acting in a way that, and producing products, proteins and metabolites and different compounds that are basically, in tune with our human pathways in a way that, can promote health. And you can try to increase, if you want to modulate your health, you can add in, if you find an organism, for example, is acting in a beneficial way, you can take more of that organism and try to see if you can, change the microbiome to even promote more of a state of health through that way. But also, again, these same organisms can also change their gene expression and sometimes act in ways that can hurt us too. So the microbiome can be beneficial when these communities are in a state of balance, but these communities are as complex as, let's say, the rainforest, right? So there's all kinds of community dynamics. They interact, they signal through, like the bacteria have their whole own chemical signaling called quorum sensing. There was recent discoveries showing that bacteria in the gut can signal through electric mediated signaling. So there's these completely complicated ecosystems, and they are just simply part of how we function. So that's what they do to help us.
I really like the way that you guys both put it regarding this is sort of their world, the microbial world. There's about 10 million viruses in every drop of seawater, actually. A lot of them are bacteriophage. Half of the oxygen in the atmosphere is actually made by microbes, not trees, but they get all the credit. You know, because they like the spotlight. But yeah, half the oxygen in the atmosphere is caused by,
no pun intended.
Yeah, half the oxygen in the environment is caused by coccolithophores, which are diatoms, and they are constantly taken out by viruses, and actually it's part of the cycle that produces most of our oxygen. So I've grown to realize that aging is really the microbial world sort of taking it back. You know, they're getting tired of the human-based gentrification of their world. So they sort of defy our barriers and get in. The big thing I wanted to talk about is microbes are definitely beneficial in the sense that when it comes to human evolution, we're actually quite lazy. We sort of evolved on top of the biological substrate that we found ourselves in as multicellular organisms. And that is especially apparent if you consider the fact that mitochondria, which produce almost all of our energy, are thought to be ancient bacteria that invaded our cells. And we said, hey, you know, you're a squatter, but we'll go ahead and let you stay here. And to this day, several metabolic pathways are entirely dependent on mitochondria. And the vitamins that we need to make those metabolic pathways work, specifically the B vitamins, are only produced in bacteria. When you buy B vitamins off the shelf, those are grown up in bacterial colonies, because it's only those older organisms that have those basic biochemical reactions that we're entirely dependent on. So there's no escaping the microbes in that sense.
Michael, did you have anything else to add to this?
I mean, all I could add in terms of, I guess, aging is that, so then, you know, the issue is why would we get out of balance with our microbiomes? And there's potentially a lot of reasons. Poor diet, just for example, poor diet, not exercising, those things will have an effect, that obvious negative effect on the microbiome, like not having enough fiber and other favorable compounds that the microbiome will metabolize, and then we'll use the end products of that for colonic health and systemic health. But I have this hypothesis that hasn't been tested. There's very little data on it, but I bet that, so if you take germ-free mice, animals that don't have a microbiome, they're not immortal, which basically says there is endogenous cellular aging. So when you consider that the microbiome is going to be, the microbiome composition, and potentially obviously functions too, is going to be dependent on the environment. For example, there are microbes that can survive in space. There are different microbes that can survive in volcanoes, in these extreme environments where you predict nothing can exist. So when you consider that, and knowing that there is cellular aging independent of the microbiome, it's probable that our cells, as they age, make different metabolites, which then fuel the growth of certain microbial species or strains that then lead to this dysbiotic state. And then you get the, you know, a dysbiotic state where the composition isn't what it should be in terms of health. So now they're producing inflammation, and there's actually a paper, Thea Varanjan, I think I'm probably butchering the name, but Don Bowdish's lab at McMaster, where they basically showed using TNF knockout mice that inflammation causes the vicious circle in terms of the microbiome and aging. So yeah, so I bet that that's what's going on. And you know, once we get into the actual fixing of cellular aging, we can actually push human lifespan out further, probably in concert with favorable changes in the microbiome. But that's like speculation. You know, that's decades of work down the line. So.
Great. So this actually is an excellent segue, what you just said, to kind of the next two yin and yang questions that I have that are relating the microbiome to aging, which is, you know, the questions that we have is, how does the microbiome contribute to aging? And why does the gut microbiome change with age? So I guess maybe Mike, you can take over, because you were starting to get on that track, and then maybe the other two participants, Cosmo and Amy, could contribute as well.
So how does the microbiome contribute to aging? I guess the best evidence would be that germ-free mice live longer. So there's something about the microbiome that actively can limit lifespan. And then going from there, it's identifying which species or strains can do these things. And unfortunately, as both these guys, Amy touched on, and Cosmo too, it's more than just bacteria that are probably part of the problem. You know, viruses being implicated in things like Alzheimer's disease and obesity. But it's once you've identified the species and the composition that's out of whack, then you can start to intervene and potentially improve health and lifespan. For example, as Amy mentioned, you can take probiotics, which are supplements of bacteria, and in some cases they can increase muscle mass, which obviously decreases with age. So yeah, I don't know. There's a big question to answer in such,
I've always thought from the beginning that the microbiome was a driver of aging. As you can see, I study more of how the microbiome drives disease processes in aging than I do look at it in health. But what's important to understand is also, you have to look at the immune response when you're thinking about the microbiome, because the human immune response is actually what kind of keeps the microbes in our body contained. Our immune system is keeping them in check, because as I said before, almost every organism in the human body, I actually did this. I put on Twitter, I was like, okay, I asked a question. I'm like, can any researcher tell me about a microbe in the human body that can't change its gene expression or acquire some genes in a way that would allow it to act as a pathogen, which is a pathobiont is the term for that. And not one researcher could respond with an organism that isn't capable of pathogenic activity under some sort of condition, right? Which is very interesting. So what it means is that all these organisms in us can actually begin to act essentially in their interest, which may be against our interest. And that can be a major driver of disease and also aging. So let's say you just start with the gut. Over time, okay, so the immune system is again regulating the virulence of these pathogens, how much they can get away with in this sense, in terms of, kind of turning against us in a way. So all the kind of, we are in a new time. All the kind of modern living stuff that we're doing, where a lot of stuff in our environment depletes the immune response, can definitely affect the microbiome. If the immune system drops because we have more chemical exposure, we have more stuff like that, where there's a lot of issues that if our systems are compromised, the microbiome will take advantage of that. The gut often becomes, in simple terms, leaky, where you have organisms that will erode essentially the gut lining. And there's actually a study which showed that even bacteriophages now can affect the lining of the gut. Essentially, it becomes more permeable. Organisms can get from the gut into the blood. That's not maybe necessarily where they should be. We're not totally sure. There seems to be, in a lot of people, microbes in the blood anyway, but more can enter the blood. There, they can get inside the cells of the immune system even more easily. Once a microbe, any of these organisms, is inside a cell, a human cell, what they'll do is get in the nucleus and actually interfere with sometimes the ability of that cell to transcribe human genes, or transcription, translation, basically the way our human cells are controlling human gene expression and the way proteins are created. Their proteins and metabolites can get in the way of all of that process, and essentially they can begin to regulate our genomes in a way that favors their survival. So of course, what a lot of these organisms do is knock down parts of the immune response, because if you can knock down part of the immune response, then you can better survive and better proliferate in a way that makes sense for you. So once we have these intracellular organisms that knock down the immune response, you kind of can get this state where, as more organisms act as pathogens, knock down the immune response, interfere with human gene expression, other pathogens can better survive. Other pathogens in the atmosphere, you might pick them up more. They kind of feed into this. They're all going to drive some forms of inflammation. That in aging is sort of known as inflammaging. Over time, people just increasingly have inflammatory symptoms, which are almost very likely driven by these organisms beginning to act up more as pathogens. And then, of course, depending on what might be going on in you, depending what microbiome community, let's say your lungs, let's say you get some pathogens in the lungs, over time you're going to see the pathogens will influence the other microbes around them to actually also begin to act sort of like bad influences. They'll signal the other microbes around them to act that way too. So over time, as someone ages, basically, I would say in simple terms, there's increasing evidence which shows that microbes begin to better act in ways that favor their survival and less of our survival. And that is a major driver of aging.
And we slowly rot to death as that happens, because we're trying to fight them off. For example, like shutting off protein synthesis, because viruses need protein synthesis to replicate. So we'll shut it off. But what's the net effect of shutting down protein synthesis? You can't make muscle. You can't make muscle cells. So
You
yeah.
both hit the nail on the head here. If you think about the selective pressure regarding these microbes as they evolved with us, sort of Red Queen hypothesis stuff. The bacteria and viruses have evolved to under a selective pressure that requires them to reproduce most efficiently in the human body, and because of that, they inhibit the immune system as part of that process. They degrade collagen, which is associated with the elastic loss of your skin that's associated with aging, and other aging-related phenotypes are also strongly associated with infection. A lot of people do not realize this. I can tell you that in the obesity field, for example, it is profoundly underappreciated that 30% of morbidly obese people are seropositive for one specific adenovirus. That is really characteristically interesting. Secondly, the accumulation of senescent cells in the human body as you age is a profound phenotype that a lot of molecular biologists at the cellular level focus on. But if you dig into the genes responsible for this senescence phenotype, the one that comes up in particular most often is the P16, P14 locus. And that gene happens to be the top gene for heart disease, which is the biggest killer of all of us. And if you dig into the virology literature, you find out that the phenotype that is senescence is actually very concordant with pretty much all viral infections, because it is advantageous for the virus to stick the cell in a state where it cannot kill itself to rid itself of infection, and it is producing all of its resources for the virus and not for the cell itself.
And I'm just going to throw a bonus in here. We've also got evidence for senescent-associated macrophages, or SAMs, as if you're familiar with Goodkoff's work. He's talking about how macrophages are being tainted in effect by senescent cell SASP, and they're also putting out more P16. So there's probably something going on there, maybe of a bacterial nature as well. So yeah, scary stuff, right? But yeah, a lot of people. Oh, sorry, Mike, carry on.
Yeah, just to add a little bit. So I see all of these companies going after aging, conquering aging by using various drugs and other things with absolutely no understanding or even an inkling of an idea that the microbiome or microbes are contributing to any of these age-related diseases, which is totally one-sided and literally half of the problem. So until you can understand the context of aging in this interaction between us and microbes, there's I hate to say it, and I hate to be the bearer of bad news, but I see most of these anti-aging therapies failing. So, and that's why the two of us, three of us, sorry, are actually there are very few people in the field who are actually, shouting to the masses saying, hey, we need to appreciate the role of the microbiome in aging. And actually, it's a big part of the story, at least half of the story. So getting out the words, so that we open people's minds to have a more broad view of aging and how we can actually make inroads on it, I think is very important. So,
yeah, I totally agree with Mike on that. Not enough people, the microbiome is continually portrayed, especially by the media, as just friendly critters in us, and we need to be looking at these microbes with more suspicion and more. We need to understand that they are always acting in their own interest, even if that happens to benefit us. It's a symbiotic relationship. But so, for example, Mike was talking about in diseases of the aging, these processes that we associate with aging. I agree. People don't realize how microbes are contributing. For example, an atherosclerotic plaque. There was a study which showed that a decent number of the lipids, I forget exactly what lipids, that become part of atherosclerotic plaque are produced by gut bacteria. So we have lipid and plaque, but it's not coming from the human genome. It's coming from microbial genomes. If you don't realize that, you start to think that all human lipid is going off or something. And when you try to fix the issue, you keep going back to the human side of what's happening. But really what you need to be doing is studying the microbes making the lipid. And since microbes produce lipids that are very similar in size and structure to human lipids, often like a kind of molecular mimicry thing, if you don't realize that the microbes are involved, you keep thinking, a lot of people just try to tie those lipid back to just human processes. And I agree, that's going to fail. You have to recognize that. The same within the brain, for example, in Alzheimer's, it's worth clarifying now that the Alzheimer's community is moving in a direction of infection as a driver of Alzheimer's. And this is really important. And so they're identifying an increasing number of different teams of pathogens that can persist in the brain. And again, there's a trend with plaque there. The amyloid beta plaque that accumulates in the Alzheimer's brain, that has always been thought of as just this negative substance that somehow forms. And honestly, I think 403 drug trials at the cost of billions of dollars have tried to create drugs that get rid of that plaque in the brain. And they've all failed. No one gets better. Literally 403 trials in a row. No one got better. And then this team at Harvard found and started doing experiments and actually showed that that plaque is part of the human innate immune response. It forms in response to pathogens that are in the brain. So I've seen this team's stuff, and they actually can show they have this neuron in a dish model that's very cool. And they'll take the pathogens that they're finding in brain tissue in Alzheimer's, and then you can actually watch videos of amyloid beta plaque growing around the viruses or the fungi. It's part of the immune response trying to control the pathogen. So you can see that a lot of what is happening in the body, we don't understand it until we factor microbes and pathogens in the picture. That's a perfect example. So now what that means is that plaque has a function. It's actually trying to stop that pathogen. Yes, the plaque may get, become too much over time and cause its own problems. But if you we need to study plaque, understanding in the brain why it's forming, and we're not even doing that. There's very few teams even looking at that right now. So we really need to factor these organisms more into what's going on.
So that touched upon a later question. I was going to ask that about the gingivitis bacteria, which was this C gingivitis? I can't remember the exact P, that's it.
Or some of the P.
Yeah, it was P, yeah. You'd think I'd know that, seeing as I wrote about this. But yeah, I saw that study where it showed that the bacteria in the mouth, which is another effectively another ecosystem aside from the gut, and it was establishing bacterial colonies in the brain of P. gingivitis, and it was like, wow, that's crazy. But it makes sense because again, it echoes the holes in the stomach or the permeability. It echoes that if you've compromised, your mouth, and you've got periodontal. I can't say the word. The gum disease, yeah.
Periodontitis.
That's it. I can't say that one. If you've got gum disease, and whatnot, it's going to open you up for deeper infection from bacteria, just the same as the stomach or the gut lining. If it's full of holes, you're going to get more bacterial metabolites and such like leaking into the bloodstream. So then it goes with the old microbial burden, doesn't it, Mike? Increases, and then that sort of starts the whole inflammation circle off, and it's a feedback loop, and it keeps going around. So the more inflammation, the more senescent cells, because the immune system is dysfunctional. It's not clearing senescent cells. More senescent cells equals more inflammation, and around she goes again. So I definitely think it's very interesting. Mike knows for a start that I've written a lot about this particular topic. It's fascinating. So there is obviously quite a lot of evidence that the microbiome communicates and also regulates the immune system. But how exactly, or how much do we know about how it does that? How much do we know at this moment in time? Probably nothing, or lots.
Can I add something to your gingivitis story?
You can, as long as it's brush your teeth. Well, yeah.
I mean, don't brush your teeth too much if you're like drawing blood every time and just making the problem worse. But I just now posted 2 images that describe the cranial nerve anatomy. So there's a whole bunch of nerves that come out of your brain and innervate your teeth, which is why they hurt so damn much. And if you think about it, that's really interesting from a point of view of a microbe that really likes infecting neurons, like viruses. So the trigeminal ganglion is this nerve bundle where 3 different cranial nerves come together. And some studies indicate that as many as 75% of human cadavers at time of death have herpes virus strains present in the trigeminal ganglion. It's profoundly common. And we know that there is a strong association between problems with cavities and Alzheimer's risk. So there's a strong likelihood that there's a route of microbes, including gingivitis, getting into the brain and causing it. And I think it might be many different microbes that do it.
Yeah, and I'll be surprised if someone doesn't mention CMV cytomegalovirus at some point, which is, Mike knows that's my pet subject, CMV, and obviously again that's a herpes-related virus as well, and a lot of people just ignore it because it's chronic rather than it doesn't immediately have an effect, but it's really quite obvious to me that the more this sort of builds up, the greater the amount of immunosurveillance is required. It's going to start having a telling effect on the immune system. It's got to. And I think the evidence supports that.
Yeah, there's actually a study done by this team in Stanford which showed that they looked at identical twins, and one was positive for CMV and one was negative, and 10% of T cells in the twin that had CMV were directed against cytomegalovirus. So you can see that a large part of the immune response is being taken up trying to keep that virus in check. And so there's less of an immune response than to start dealing with other issues that might be happening to the body. So that's just one way. These persistent pathogens are continually, almost keeping the immune system, forced on them, as opposed to being able to focus on what they should also be doing to keep people in better shape and not age as much. So that's a good example. And all these pathogens, as you say, when you're talking about the immune response, they've developed a lot of common strategies often to disable key parts of the immune response, because of course that can allow them to survive. And part of the early research I did, we looked at this receptor in the human body, the vitamin D nuclear receptor, actually, VDR. And we showed that HIV, CMV, this was from different studies, but we pulled it together, Borrelia, different fungal species. All of these organisms have evolved to either themselves or create proteins or metabolites that can bind into that receptor and slow its activity. And the VDR plays a very important role in controlling the innate immunity. It expresses antimicrobial peptides. It helps express TLR2. So think about it. If you're a virus and you tone down how that receptor can function, you just tone down these key parts of the innate immune response. Now you survive better, but the host is immunocompromised. Now that person is more likely to, because they're immunocompromised, pick up maybe even another pathogen from the outside world or whatever's going on. That pathogen will also probably survive by slowing the immune response. Now the immune response drops again. You can kind of see this situation where it becomes like a snowball rolling down the hill, where the more organisms you acquire, the more the immune response drops and the more pathogens are able to act in ways that begin to age the body, in simple terms.
Yeah, and that also ties in with another news article again this week. I talked to a lot of stem cell researchers, and they managed to, you may have seen the story, they managed to confirm that they cured HIV in a second patient using bone marrow transplant, which effectively reboots the immune system, really. And it's long been proposed that, certainly by some stem cell researchers that I know, they're saying if we could effectively rejuvenate the actual the blood producing stem cells and the supporting mesenchymal stem cells in the bone marrow, they think that could greatly alleviate the situation from the top down. So there's good evidence for that. So it really is complex, but I think it's fixable. And rejuvenating the bone marrow strongly suggests that we can do something about immunosenescence. So, what are your thoughts about combating that?
Yeah, no, that's a good example with stem cells. And some people, there's even talk that stem cells themselves may become infected, which seems logical to me over time. And so if you think about it, if you're in the most simple terms, if someone's dealing with a growing infectious pathogenic burden that's wearing down their body, and you bring in an influx of new, healthy, non-infected cells that can begin to, act in a better way, that can inhibit the growth of some of these organisms. There's also a couple teams teams who doing early immunotherapy, in which you can actually activate or sort of stimulate the immune response to better go after some of these pathogens. There was just a team that did that with HIV, really early study, but they actually did immunotherapy against, where they looked at a protein, I think, that HIV was creating, and they actually had the immune system created a treatment that, or we're testing whether the immune system itself could recognize the virus better and target it over time. So we can also not only, what I mean by that is we don't have to just create interventions that affect the microbiome itself for aging. We can also create interventions that improve the immune response and allow it to better fight back against this growing microbial burden. And introducing new, fresh, uninfected cells would actually be part of that. So it's a good idea.
Yes, I mean that's also building towards what some people talk about, engineering resilience. So not only are we, thinking about the idea of repairing the damage periodically, we're also thinking about making our cells more robust. So if our T cells and our macrophages are better at hunting out these pathogens and destroying them, I mean,
we can improve.
And it could also have a knock-on effect with senescent cells. I know a number of people who have proposed that the ultimate senolytic wouldn't be a small drug like they're trying now. That's more of a stopgap. They see that rejuvenating the immune system or enhancing the immune system would be the best form of senolytic, because it'd start clearing house without the need to use drugs at all.
Right, because think about it, the immune system has evolved with these organisms since the beginning of time. So it actually has defenses that are very good in terms of controlling pathogens. If we can enhance those natural defenses that the immune system has, that's a really cool way to move forward. So I agree with you there. And then also, you have to think about how we can use this in a preventative fashion. If we can create treatments that keep the immune system in better shape earlier in life, we may prevent pathogens from being able to even act in ways that age people over time. So we could actually, what I think we need to do most is actually keep the immune system in good shape, starting even early in life with therapies that prevent it from being overrun by pathogens that can take it down.
Yeah.
Just to add a little bit, so I'm guessing there will be a map of the human body and the microbes that can inhabit it in various conditions, whether that's aging or some disease. But if so based on the physiology of a given organ and how that organ decays over time, the microbiome is going to reflect how that organ has decayed. So for example, maybe in the brain, herpes viruses can better exist in that environment, or other viruses, whatever it may be, because I know that bacteria and fungi have been reported in the brains of Alzheimer's disease in the A-beta tangles. But there's going to be a limited range. It isn't going to be thousands of species. Oh, that's my bias. That's my bet. But for each tissue, that will probably be different. And once we've mapped every tissue, including the immune system, like what are the microbes from a polymicrobial point of view, not just bacteria, but viruses, fungi, archaea, whatever, and all of them. What are the ones that can negatively affect the immune system, and then all the other organs. And once you've got that map, then you can go and not just immunorejuvenate as his broad term, but you can selectively go after, targets in each tissue, selective targets in each tissue, in addition to the immune system, to really maximize the health approach. But that's going to need computer modeling and all kinds of advanced tools that I don't think are anywhere near to use right now, unfortunately. So,
and touching upon that, the harmful bacteria, to talk about the beneficial or at least not as harmful bacteria, what about the role of bacteria that are creating, the various short chain fatty acids, like butyrate or butyrate or whatever you want to call it, butyrate I call it, that they feed the colonocytes I understand about 70% of their ATP comes from those particular bacteria breaking down and creating these fibrous materials and creating these short chain fatty acids. What do you think to the idea of trying to enhance or increase the populations of beneficial bacteria as a strategy? Any thoughts on that?
You can artificially increase the number of butyrate producing bacteria, but they would need the substrate, which in most cases should be fiber. But then you know, you can get a massive debate on how much fiber is going to be optimal. For some people, maybe it's 100 grams, which is what I shoot for a day. But for other people, maybe they can get by with optimal butyrate producing bacteria with less. But again, you'd have to figure that out. But the way I see that is, think about lifespan, and you think about athlete lifespan, and you think about the average person lifespan. Athlete lifespan is just shifted up. You know, they live about 15 to 20 years longer, but you know, they're not immortal. So maybe you maximize butyrate production, but the fact is, you're like on an athlete, so you're basically on an athlete microbiome in terms of butyrate producing bacteria. So, you know, you could have the best diet, but eventually the barrier is going to get leaky because there isn't enough butyrate or enough short chain fatty acids. Because as Amy mentioned, you know, these buggers have evolved to exist to wear down our immune defenses and to wear down our epithelial cells to leak into places that they shouldn't be. So, you know, we're going to need advanced therapies. And I hate to plug the book, but I mentioned this, you know, that, you know, 120 plus years ago, mortality was sky high. And one of the reasons that we, you know, possibly changed that was vaccination. And, you know, Amy and I talked about this a little bit already, but, you know, we need tools like that can you know, help older people to react better to microbes and microbes that affect us during aging and disease. So whether that's vaccination or immunorejuvenation or whatever it may be, but more scientists need to be studying this, and unfortunately, it's, as I mentioned, it's understudied.
Although there has been some recent progress on that front with mTOR inhibitors. There was a phase 2, I believe, which showed positive results for response to flu. And that was the TORC1 pathway, I think it was. I can never remember which it is, TORC1 and TORC2. And I think TORC1 is the bad one and TORC2 is the good one. So they've Rebooting managed to actually inhibit the one pathway out of mTOR that was detrimental, and it seems to improve the immune system, a bit like rapamycin, except not as harsh. Any thoughts on mTOR inhibitors at all, guys?
So I think there is a little bit of data that microbes can actually activate mTOR, which so then inhibiting it would seem to be a good thing. But as you mentioned, I think there's also data showing that mTOR is involved in the immune response. So it's a tricky fine-tuning rheostat. And what was you mentioned mTOR, and what was the other?
Well, there's two pathways, isn't it? There's TORC1 and TORC2, and I think it's TORC1 is the bad one and TORC2 is the good one, and they've
rapamycin.
Yeah, they found a way to inhibit the one that they don't want, but allow the normal function of the one that they do.
Right. I think when it comes to microbes, it's you know, so antibiotics, which were great for 100 plus years, now we're finding antibiotic resistance is growing because microbes evolve. So eventually we're going to have more accurate tools, computer-based tools that will predict weak sites in the microbes that are affecting us, and will be more targeted approaches to affecting to attack them basically. So when it comes to mTOR, or when I hear rapamycin, I think of these as the broad tools, the antibiotics, the rough tools that'll help you a little bit, but are they really addressing the problem? I think we've got to use genomics to identify who's there and in what conditions, and then what are the specific, like I said, computer modeling based approaches that it can attack them and destroy them completely while barely or almost not affecting our cells at all. That's the future that I hope to see. Yeah.
It certainly sounds like a challenge, but I don't know. My money's on engineering our own immune systems to be better. To be honest, immunotherapy I think is extremely promising, and I think we could make some indomitable macrophages and much more robust T cells. And I've even seen some experimentation where they've taken non-immune cells and given them immune cell-like abilities, which is very interesting indeed. So it's intriguing because they're looking at doing that to hunt down cancer. So the cancer doesn't recognise it as an immune cell, and it spots the cancer. So very interesting. So yeah, I mean, there's plenty of things that we can work towards to ultimately bring that under control. But I'm sure a lot of people here are probably wondering what we can do now. And I know I've got a feeling Mike's going to pretty much tell us what your granny would have told you about sleep, exercise, and a decent diet. So with that in mind, what is, and this is a very broad question, what is the ideal diet or kind of diet that may encourage a healthy gut microbiome?
I don't know, maybe Amy and Cosmo want to chime in, because you know me, I can go off on this for a long time.
I've got nothing but shitty ideas.
I think that this is one of the areas where personalized medicine is going to matter the most. I think Mike was alluding to that. We're going to have to, like he said, get a better idea. Each person is going to have to understand their microbiome better, what organisms are persisting in them. And again, I usually refer to them as dominant pathogens, the organisms in those communities that can cause the most trouble and sort of influence the microbes around them to also begin to act in more troublesome ways. So it depends on the person, especially because the way you eat. And so I work with a functional medicine group, and you can see that it really depends on the case. If the fiber can be good, for example, of course it can be. Microbes can use it as a base for things that are beneficial. But for example, if you have Crohn's disease, you have a microbiome that is very dysbiotic already, so very imbalanced. Pathogens have taken over. There are very few microbes acting in your interest. You don't necessarily want to eat a lot of fiber at that point, because you don't want that microbiome community to flourish. You actually want to change that microbiome community into one that might be less imbalanced and more healthy. So there, you'll actually see protocols where we pull fiber first. We pull sugar out. We pull anything that would be feeding or helping those organisms out. And we do kind of a kill phase where we use substances that would target pathogens that are in that community. Over time, as that begins to happen and hopefully pathogens go down, you can use interventions, some other stuff, possibly probiotics, to begin to build the microbiome into better shape. At that point, you might want to begin to add fiber back in and stuff that then begins to support those organisms. So what you eat depends a lot on the state of the microbes in your gut and where they are. If you like what you have, you want to keep it, and you want to eat in a way that preserves your microbiome. If you're not happy with your microbiome, you may want to eat in a way that can change it up, right? So it really is going to depend on the individual patient. Go on, Mike. I know you have more to say.
And just on, hang on, Mike, you're muted there. Just on top of that, though, just wanted to add a secondary question. What happens if you've predominantly eaten a very bad diet and you've got the sorts of bacteria that you don't want? Is there a point of no return, or is there always going to be a small population of beneficial bacteria, that eat fiber and what? Or is it possible to completely damage your microbiome to the point that you can't come back?
Oh, no. I think that the body is very resilient in a lot of ways. And I think that if we, honestly, I think that also you want to consider the immune response. If you try to improve the immune response and then you try to improve the microbiome over time, I mean, in simple terms, there are protocols people are already using that sort of starve pathogens, especially fungal species, different things like that, where you actually, for example, sugar. It's a simple thing. If your microbiome isn't in good shape, pulling sugar is probably a good idea. A lot of pathogens use glucose as a source of energy. So you actually try to remove energy sources and other things, substrates that would be feeding pathogens. But if that can work all the time. I don't think there's such a thing. Well, I don't want to say that, but there might be very difficult cases with the microbiome, but I don't think that we should have an attitude that you can't improve it. I think that, and we're going to need more interventions and more targeted approaches, but you can bring stuff back. You can, the body wants to, whether it's the immune response or the microbiome, in a way, as soon as you give the immune system or the body a little bit of an edge against the pathogens, it will run with that. That's what we see often in patients. So it's encouraging because you can change things already today.
Yeah, I agree with that 100%. And also the personalized approach that Amy suggested. So I wouldn't, even though I think a very high fiber diet and not the 30 grams at the RDA or, is recommended, 100 grams or so. Although I'm all in favor of that, to recommend that to someone who's got Crohn's, who's got, irritable bowel syndrome will be, stupid. So that's why it goes back to this personalized approach. But then even still, what is the ideal microbiome? Like to even say, all right, you need to have this amount of butyrate producers and this amount of that producer and this amount of biotin or whatever producers is nonsensical. So you would need to have your microbiome composition in addition to circulating biomarkers or other markers of health. And then once you see that, then you could potentially play with your microbiome to see if you can tweak your markers of systemic metabolism. But if the goal is to improve the short chain fatty acid production in the gut, there are things like exercise. I mean, it's like you said, things that your granite suggested. Exercise increases the butyrate producers. Athletes have higher levels of butyrate producers. And if I don't know how familiar you are with athlete diets, but I know a few, the diets are terrible. So even with a terrible diet, a low fiber diet, squirting glucose packs, which aren't going to be beneficial for your microbiome at all. As Amy mentioned, it's sugar. It's probably going to feed, things like candida or whatever else. They've got terrible diets, and yet because you can exercise so much and have a high level of these butyrate producing and other short chain fatty acid producing species. So again, exercise, a high fiber diet, lots of polyphenols, things like berries. But then again, it goes back to quantifying your biological age, and using tools like Alex's aging.ai can help with that, where you're using the systemic markers of health to see how well your diet is affecting your microbiome and your metabolism. Or Morgan Levine's, stuff where she's got the nine biomarker panel. So the biomarkers in concert with the microbiome, and then using your diet to tweak that. That's what I do. So if everybody did that, I bet their health would be much better.
Sorry, I want to say something. I think it's also worth noting that we need to think a lot about the inherited microbiome. So it's like, where does our microbiome come from? How does it start? We inherit a lot our organisms from our parents, mostly the mother, right? So not that it's been just a couple years. We know, for example, there's a breast milk microbiome, right? So that's important. If there's this very robust microbiome in breast milk, and there's also a lot of immune compounds in breast milk, like lactoferrin and antibiofilm type compounds that are in the breast milk along with these organisms. Now think about that. If an infant is formula fed, they were never given these, most mothers, some mothers breastfeed for years, right? So they're missing years of this influx of microbes that would come from the mother normally, and the immune compounds that help these microbes better survive and persist, I would say, in an infant. So we have to think about those practices a lot too. Now, you don't want to always think though that the breast milk microbiome is perfect, because the health of the mother will influence the composition of the breast milk microbiome. So there was actually a study which showed that obese weight mothers had a less diverse and more dysbiotic breast milk microbiome. So in some cases, and I guess here we can share some ideas. I've always thought, for example, that we want to think about things like, let's say there was an obese weight mother or something. We realized already we could test her breast milk. We realized there was already dysbiosis and less balance in the microbes she was going to give to the infant. We could have that mother possibly, the infant, we could supplement with milk from possibly a healthy weight mother. That's something I've always considered. Maybe there can be a milk bank, and you could go to the milk bank and say, look, I have these issues where I'm lacking these organisms. Can you give me milk from a donor that might have those organisms or immune compounds? We can begin at that stage to intervene, because if we set up the microbiome in infants where it's already dysbiotic, or imbalanced, we're starting people off at the very first part of life with trending towards a path that's going to cause disease and more inflammation. So we need to begin with literally, and also there are studies that show that organisms can persist in the womb, and we need to better study that. I am frustrated by the fact that there are not more teams studying organisms that can seed infants in the womb, because if you think about that, if there is a placental microbiome, if there are organisms in the amniotic fluid, which there often are, they will seed an infant again, starting in the womb. And those can be pathogens. And if you start off with pathogens already in these microbiome communities, you're immediately starting life in a worse place than other people. So we really have to think about the maternal microbiome and inherited organisms too.
Just to add a little bit to that, Amy, so there are actually transgenerational effects of eating a low-fiber diet. So in a study done in mice, and I'm sure you've seen that, where they gave a low-fiber diet, and then it turned off genes, and then in the next generation, and then they had a low-fiber diet, and it kept turning off genes so that, I don't remember how many generations later, but genes were completely turned off. You couldn't do anything. So beyond just the effects of mother offspring, I mean, you know, and then you think about the, you know, the standard American diet. People are eating these heavily processed foods that have no nutritional value at all. They're probably, you know, imparting that on their kids, and there you go. So.
Right. So you need to start from day one thinking about nutrition, about these things, and not wait until you, I mean, the future is not going to be waiting until people feel really sick and go to the doctor and already have a bunch of symptoms. You want to, I think we want to begin in the womb sometimes, day one. And,
or before. Right.
Exactly. With the mother's health, literally modulating the parents' health in a way that can help, produce a microbiome, that will seed the infant. Yeah. Really need to think about those trends too. And also, for example, another example of that, which you'll see mentioned more, is cesarean section versus vaginal delivery, which I actually think the breast milk microbiome has the biggest impact on an infant. But for example, there's a vaginal microbiome. So if an infant is born vaginally, the infant goes through the birth canal and acquires on its skin when microbes from that vaginal microbiome seed the infants, the first microbes on the infant's skin. If there's C-section, you take the infant out, they don't get those microbes. What they do pick up instead on their skin, there's been a couple studies, just the microbes in the operating room, which can be really weird and can be some serious pathogens. So you have these infants that are seeded when they're born with surgery room organisms, as opposed to what should be coming from the mother. There's topics like that we need to think about more. So we need more advocacy. I understand if people have an emergency C-section, but we do a lot of C-sections just for, in the US, just because, say, hey, do you want a C-section? No big deal. And the mother says, yeah, I prefer a C-section. No, we need to begin with more education that is going to affect the microbiome, that can affect infant health. We need to be emphasizing that more.
I think I read that there are vaginal swabs on babies who have been born by C-section. So they'll swab the mother to try to get some of those microbes onto the baby.
We're trying that, exactly.
Yeah. I just sent the link in the chat. Rob Knight actually did that. He's the guy that runs the American Gut Study down in San Diego.
Nice. Did it to his own child. Yeah, he did a TED Talk where they took microbes from his wife and just coded their infant when the infant was born by C-section. Yeah. So these are all things that we can think about right now, and most people are not thinking about them though.
It's like a bacterial baptism.
Yeah, that's
way more useful.
And real. Right?
I gotta add into some of the milk story that you mentioned. Especially since my last name is Mielke. We got to talk about milk. So, fun facts about milk. Did you know that human breast milk actually has sugars in it that cannot be processed at all by the human body? In fact, those specific sugars can only be processed by Bifidobacteria infantis, which is the species of bacteria that we find in infant guts periodically or frequently. Another interesting thing about milk, aside from seeding the bacterial populations that we want in the newborn infant, milk also has a lot of molecules in it that are viral decoys, specifically sialic acid. So milk is loaded with sialic acid, and sialic acid is what various viruses, especially influenza virus, uses to get into cells, right? So if we think about cellular senescence is primarily a phenotype that we define by P16 upregulation that halts the cell cycle. But the thing that aging biologists consistently use to study cellular senescence is an activity of a specific enzyme called a senescence-associated beta-galactosidase. Now, they found that activity elevated in senescent cells a long time ago, but only recently have figured out what that gene is, and it's the GLB1 gene, and that's the gene that cuts the sialic acid off of the cell membrane in human cells, because all of our human cells have sialic acid on them. So there's a direct connection there with virology and the way that viruses get into cells. So it's thought that mother's milk is predominantly providing that viral decoy by clogging up all the binding sites and viruses so that they pass through the gut without entering the epithelial cells.
That's very interesting.
So could you make the argument that past a certain age you shouldn't be drinking mother's milk, because then maybe I don't know, see, I'm with you on the sialic acid, and you're feeding the growth of things like bifidobacteria, which is a good thing for the babies. But then I'd imagine that if you kept drinking the mother's milk past a certain age, maybe you'd start to feed pathobionts. I don't know. I'm guessing nobody's even looked at it.
I think after about age
43,
30 or so, you don't want to drink mother's milk anymore.
43, you're definitely not appropriate, Mike. So stop that.
Yeah, follow nature. You know, I would think you could do that, but I would say that maybe we should just try to make what nature normally does most beneficial. If people usually breastfeed for early years, let's get that breast milk in good shape. Let's get the immune compounds in there. Let's make sure this is happening. If we create formula, let's try to create a formula that might even have less semblance to breast milk. Formula now is got to be, there's almost nothing in formula now that we know that's beneficial for infants. So we need to, if we do create an artificial formula, we could revolutionize formulas. We could take some of these antimicrobial compounds that are in maternal breast milk and at least add them into formula or something like that. But that's not on too many people's radar either. So, or
just powdered mother's milk, right? Instead of powdered formula.
You know, exactly.
Highly recommend, especially when snorted. Sialylated lactic acid is or lactate is the primary constituent in mother's milk that is highly sialylated. That's where most of the sialic acid comes from, and it's really expensive if you buy it from a lab source. But I would love to find some supplemental form of sialic acid in particular.
Yeah.
Those are stereotypically good microbifidobacteria. For example, it's like 40% or something, some high number in babies, and then it declines almost to 0 in older adults. What's interesting is that I'm completely formula fed. My mom didn't breastfeed me at all. So I wonder if my interest in the microbiome ties back to that, not having that, you know, as a baby. So
tell me about your parents.
Quick question again. This correlation with the altered microbiome and aging. Has anybody done a study? Because everybody's been looking at microbiomes in mice in a variety of different contexts. You know, old, young. But with regards to fecal transplants, has anybody tried? Maybe there was a study done. Tried taking, the gut microbiome, transplanting it from a baby mouse or young mouse to an old mouse, and vice versa.
So as far as I know, there are only 2 studies that have done fecal transplant from old into young. One of them is in mice, and they basically saw what you would actually. There are 3 studies. Sorry, one is old into young, and it goes exactly like you would predict. The young mouse exhibits some of the old age phenotype. In a second study, they took old poop and put it in young germ-free mice, and the young germ-free mice didn't actually take the bacteria. Like after 1 week, it ended up looking like the gut microbiome of a young mouse. And then in the third study is actually my data. We did it in humans into germ-free mice. And like I said, that data is now in review. It's been in review for 2 months, man. Who knows when I'll get something back on it. But we were trying to see if the old age phenotype in high-functioning older adults versus lower-functioning older adults could be transmissible through their gut microbiome, with the prediction that higher-functioning older adults who had a better body composition, more % lean mass, less % body fat, and better muscle strength and other measures of physical function. So the groups are different for that. So we put that into these germ-free mice, expecting to see better functioning and less functioning germ-free mice. And actually, there was no transmissible effect of the gut microbiome on body composition, but muscle strength was increased in the high-functioning mice, which suggests a causative role for the microbiome on muscle strength. So, but there needs to be more studies. And I don't know that the germ-free model, fecal transplants into the germ-free model are necessarily ideal, because in theory you should have, aged germ-free mice taking poop from aged human or aged mice donors, but that's very expensive to age, mice for 2 to 2.5 years to do that. So,
yeah, it's very hard to control for all those factors.
But Dawn Bowdish's group, they did some beautiful work on that. If anyone wants to look up that study, I think it's Cell paper last year or the year before. Bowdish DA. That's the PubMed search.
All right, Steve, where are we at right now? Unfortunately, I just barreled in here in the middle of
a discussion about milk. Yeah. Well, I mean, it sort of broadly leads up to the next question, which I was going to ask, which is, what are your thoughts on probiotics? You know, we're constantly bombarded in the supermarket here in the UK. I'm sure it's the same in the US with yogurt drinks and Yakult, and not that I'm endorsing a particular brand here. You know, probiotic yogurt, everything's probiotic, everything's active. But what, I mean, is it really any use?
So yeah, I'd say the first step in taking a probiotic would be seeing what your gut microbiome composition is in the first place. But then you would also need your circulating biomarkers because, you know, I think it's essential to have both. But just as an example, I don't have or have almost never had any bifidobacteria going back, probably because I wasn't breastfed or I took a lot of antibiotics as a kid, which are known to kill bifidobacteria. And because they're associated with youth, I don't want to have the bifidobacteria of an 80-year-old. So I've tried to add them back in using a probiotic supplementation approach. So the first 2 times I did it with a relatively low dose and they both failed. So, but I know that it failed because I actually tested my microbiome. I'd say most people take probiotics and it's faith-based. They think that it's doing something and they have no idea if it's even in their gut. So the 3rd time I did it, I increased my bifidobacteria from 0% to something like 1% of total. And I saw some actual positive changes in my sleep. I've written a post about this after the fact. But with that in mind, I try to supplement with a little bit more bifidobacteria. And that amount of bifidobacteria maybe is not good for the rest of my gut because I started to have some GI issues that I didn't have before. So as soon as I stopped that, they went away. So I'd say a targeted approach for probiotics is essential, and monitoring not just your microbiome, but other markers of systemic health, because I think there's some emerging data too that probiotic supplementation can actually induce bad things. So there's an abundance of literature on probiotics in various conditions being beneficial, but it's context dependent. It depends on the state of your ecosystem, the state of your microbial ecosystem down there. So,
hey, Mike. So I suspect that the bifidobacterium that you're taking as a probiotic is getting eaten by your stomach and your duodenum in particular. So there might be a way around that.
So there are certain coatings that you can get around the acidity of the stomach, but they're in there. I've increased them, not by much.
I meant supplementing through the other end. But
I've actually tried that. It didn't work. I've tried using it going in the other end.
So you've done a yogurt enema, yeah? Okay.
Not yogurt, actually just the pills themselves. But
so, fun fact: the only known treatment for obesity in the medical field is stomach surgery, specifically Roux-en-Y gastric bypass. That's the only provable cure for obesity. And the way that we've recently figured out how it works is it actually allows microbes that you're taking from your gut to bypass the duodenum, get past the small intestine and go into the jejunum, the middle intestine, where the pH levels are not such that they destroy the bacteria. And as a consequence, the diabetic phenotype within stomach surgery patients resolves within a week, and the gut bacteria diversity goes way up.
So I'm hearing a lot of I guess proactive measures that we can be taking to improve our gut microbiome health. But I'm listening in on this, and I think this is really cool and all. And I'm kind of jumping to the later questions here. Is okay? How do I do this? How do I measure my gut microbiome? If I'm taking I go to a store, I buy a probiotic, or maybe I want to measure my gut microbiome before and after. Where do I even begin? Do I go to my general practitioner and say, hey, I'm going to self-experiment. You're in on this project with me. Or maybe that's not the best way. How do I do it? I mean, I don't even know where to begin. I mean, I can Google it. I can look it up, but we have a panel of experts here. I want to start trying, I want to see if, let's say, drinking lots of yogurt is going to be beneficial, or taking some other type of probiotic measure is going to be beneficial. You know, some of the questions we have here is what commercial methods are available for measuring our microbiome health? What biomarkers can we use at home to monitor our microbiome? But I guess more broadly speaking, you know, how do I take control of my microbiome health, and how do I go ahead and measure it, and how do I, you know, get more educated as far as whether a certain product is working? How do you do that, practically speaking?
So there are at least two companies that I have off the top of my head that use different methods. One of them is 16S ribosomal DNA sequencing, and the other is basically looking at the expression patterns, mRNA. So the first company is Ubiome. It's about $90. They use the 16S approach, but the 16S approach is not very good below the genus level. It gives you a few species, but does not give you strains. Ideally, you would want whole genome sequencing, but as far as I know, it's not commercially available, and that would give you actual species and strains, because not, as Amy mentioned, you know, things that are that can be thought of as commensals can become pathobionts under certain conditions. So knowing the exact strain is important, especially for probiotic-based studies. And then the other company is Viome that I know of, that's using the mRNA sequencing-based approach. I use Ubiome. That isn't to say it's better or worse. I've done Viome too, and there are probably other companies that are commercially available to study the gut microbiome. But those are the first two that come to the top of my head.
Well, and the American Gut Project, run by Rod Knight in San Diego, right, which has I haven't done it, but I think they were the first on the spot to do this type of thing, and they're strongly rooted in open source and community science, so worth looking into.
Do you just send?
Go ahead, Mike.
I was just going to say, do you just send a fecal sample to American Gut? I don't know how that even works.
I think so. Yeah, I remember talking to Rod Knight at one of the conferences, and then talking to some of his lab mates, and they all told me that there's like a not so friendly relationship between them and Ubiome, because Ubiome is for-profit, closed source pipelines. American Gut, though, their entire analysis pipeline is open source. You can go scrutinize it. I think to answer your question, Steve, I think that the best way that we can move forward in what to do about the microbiome and figure out what products are best is to get a substantial number of people out there who have done Ubiome to release their data. And you can do that through platforms like Open Humans. Because we need to aggregate all that data together and also combine it with information on what probiotics people are using, because as it stands, it's still too expensive to really do it that frequently. You can't really measure your microbiome daily. Maybe someday we'll have sequencers that are cheap enough to do that, but until then, we're entirely dependent on a large cross-sectional study to find any answers.
Right. I think also this topic is sort of like the hot question at the moment. If you can answer, how do we do this? You would be a very successful company at the moment. I'm regularly called by biotech companies that are trying to find people who want to do this. And in fact, I just started working with one. So I'm literally going to be working on developing their testing. What I think a problem is that we're not including viruses in these analyses, especially phages. So remember, as I mentioned before, bacteriophages are viruses that infect bacteria and modulate their behavior. So you can do a whole lot of testing and look at bacteria, but if you don't realize that bacteria, the activity of that microbe is being modulated by a virus in its environment, you're not going to understand what it's doing. And a good example of that is there's, for example, a small study, but I think it's interesting. And as we were talking about before, microbes in the gut produce neurotransmitters, chemicals that we as humans can use. So for example, there are certain bacteria in the gut that produce dopamine. But a recent study that looked at patients with Parkinson's, small sample size, found that there is a specific bacteriophage that infects those bacteria that produce dopamine and modulates how they create dopamine, right? So you could measure dopamine all day. You could measure the bacteria that produce dopamine all day, but if you don't bring that bacteriophage into the picture, which is the driver of how dopamine is being produced and regulated, you'll miss a lot. So one of the challenges is going to be creating testing that brings the viruses in these communities into these analyses. I don't think Ubiome has that yet. And also increased testing, I think, that also looks for microbial and viral proteins and metabolites, because it's not just the microbes, it's the compounds that they create that become important in affecting human pathways. And so what's good about that is looking at proteins and metabolites is there you can pick up on more common trends, because there are certain different types of organisms that all make similar compounds. So as opposed to having to try to get into the nitty-gritty of every strain of every organism, which is almost impossible, or you'd need a huge amount of the computing power would have to go up dramatically. You can say, I think I need more of this compound. There are certain types of organisms that produce this, or there's a lack of this metabolite or a lack of this, and you can go off bigger trends that might be able to help that. So I think we need to measure those proteins and metabolites better in these analyses so we can go in that direction too.
Just to add a little bit to that. So I'd like to see it where we can get the entire ecosystem, not just the viruses and bacteria, fungi, everything that's down there, and mapped. But then just along the lines of the proteins and what other metabolites they're producing. So even if you have that map and you have what they're producing, if you saw something and you're like, oh, these microbes are producing these metabolites, we need to get rid of them. Ideally, also, you'd want that picture of systemic health too, because even if some certain microbes are producing certain metabolites or other stuff, if we're able to handle it and our systemic, biology is fine, then not a problem. But ideally, you would want that snapshot over time, because then you could say, all right, this is what my microbiome, my full picture of my microbiome looked like in health, in optimal health. And now 5 years later, it's this. Now you've got to get back to where you were or should be. So, even going to your point about in the womb, having measurements maybe every year, your GP when you go to the doctor, and having tracking that over time is probably something we should all.
Right. Definitely tracking over time is so important. If you do just a sample at one point in time, it gives you almost no information. And there's so many, the difficulty with the gut is there are so many variables that affect the gut microbiome. It's a little insane. In fact, Bob Knight, we're talking about, he published a paper a while ago called Moving Pictures of the Human Microbiome, and that made a big impression on me. I mean, he showed just one thing you eat can change the way the test comes back. I mean, depending on women, if they have their period, just different variables that where you're at that day. There's so many things. So I also wonder if we should also be, as you're saying, I think we should look in the blood too, for example, because organisms that get into the blood are going to be most likely bigger drivers of disease, probably more dominant organisms that are influencing these communities. So we need to test outside the gut too, because it's going to be organisms and the proteins and metabolites they're creating when they leak out of the gut or in the blood that would indicate possibly some of the bigger drivers of this. So I think we need to also get into areas of the body that have less of that noise. And by that, so I'm also interested in blood testing for organisms too. That could be done, more commercially for patients.
Brilliant idea.
Full sequencing though, because like right now you could, because right now.
2 companies I think that are trying to do full sequencing now. I think one,
Viomer, French company, or I don't know if they're French, but it's Viomer, V-A-I-M-E-R. I don't know if it's full sequencing, but I'm pretty sure they do at least the bacterial component.
It's hard to keep up, but there's a couple I think that at least made it their goal, but whether the cost is going to work out and all that is going to be a challenge. I'm pretty sure we'll get there. It's just a matter of when. Yeah.
But like most scientists measure LPS, lipopolysaccharide, but that's only a marker of gram-negative bacteria, right? You've got gram-positive, you've got fungi, you've got viruses, and then you've got DNA viruses, RNA viruses. You have to.
Right.
So.
Yeah. And I guess also testing that identifies what I will call, again, dominant pathogens. For example, I really think we do need to focus more on pathogens, not just on what could be good microbes, but what the microbes are that are actually driving dysbiosis. So, example, if you look in the mouth, and there's a study I love, it's a tooth, and it just shows the organisms in the oral microbiome as the tooth moves towards periodontitis. And you can see basically P. gingivalis that we were talking about before that was just found in the Alzheimer's brain. P. gingivalis is it's an instigator. That's how I would say. It acts up. It starts changing its gene expression. It starts excreting virulence factors, signaling, quorum sensing. All these organisms around it go, oh, and it's they start to change their genomes and act more as pathogens. And then this study actually tracks the gene expression in these teeth that are becoming, you know, more like cavities in a simple sense. And by the time that the periodontitis forms, even the organisms that are considered to be the most innocuous organisms are producing virulence factors. Wow. So if you think about it, let's say that was a society. It would be sort of like if a dictator came in, right? And all of a sudden the whole society is changing based on that ideology. If you can get that dictator and kind of get that regime out of the way, that can go a long way towards allowing the body or the community itself to restore. I see the same trend in the human body. If we can better identify these dominant pathogens that drive dysbiosis and target them too, then the body and the microbe community may itself rejuvenate in a way that's going to actually rejuvenate in ways that we maybe couldn't even predict as humans. Because of course, if it happens naturally, then it's going to happen in a way that might even be most accurate. And that's kind of cool. So I'm all for pathogen testing as well.
That's a very interesting concept, targeting the pathogens that drive the initial, you know, infection within a community. I think we just covered huge amount of topics under the umbrella of microbiome and aging. We've been asking questions. Steve has been asking questions. I've been asking questions. The panelists have been asking questions between themselves. And we've been basically taking some questions in the chat. But right now, I'd like to kind of do a little bit more formal shout out to everyone who's listening. And people can either use the either in chat or use the raise hand. I guess there's a raise hand feature. I didn't even know that. If they wish to use the mic, Nicola and Josh will moderate. So I'm going to step back a little bit here and open the floor to folks that have burning questions to ask our panelists regarding the topic that we just discussed. So do we have a list, Steve, or are people going to just kind of pounce in right now?
Oh, I mean, how
here we go.
Oh, okay. Elena's already got a question.
Questions are flooding in.
I can read it this time. Can you please explain the relationship between leaky gut, microbiome, and physical activity? That's a very good question.
So that can go two ways. One, like as I mentioned, physical activity and exercise can increase the butyrate-producing bacteria, some of these short chains that are important in gut barrier function. So, but then alternatively, there's also data on extreme exercise, like I think it's 24 hours straight or 48 hours straight. Where it's just you're running for that long, and it induces leaky gut. So there's probably a fine line of how much exercise is good and how much is not in terms of leaky gut.
And what exactly is leaky gut? Is it just like the name, the bindings between the cohesion between cells is permeable, and you basically have fluids from the lumen leak into the bloodstream? Is that correct?
Fluids and metabolites or DNA, RNA, the tight junctions in between the epithelial cells become dislodged or detached or aren't expressed or whatever, and the stuff can freely pass between the lumen and the interstitial space leading to the blood.
Right. Although there is some permeability in the gut always. I mean, a decent number or a good number of the metabolites just in blood and even a healthy individual are derived from the gut microbiome. So we need to take that into consideration too. I mean, even in healthy people, what's going on in the gut ends up in the blood a lot. Of course, in disease, it's basically just an exacerbation where the gut lining becomes more permeable in a sense. So maybe organisms that wouldn't normally go from the gut into the rest of the body would get that more. But, that's kind of part of it too. I think, that's exercise though. Yeah, that's more your thing, Mike. I think one thing that I think is over exercising, like you mentioned, I think that's a caution actually. There are some studies which indicate that if you push yourself too hard, it might actually be detrimental. So I'm not definitely advocating for everyone to become a crazy marathon runner necessarily. And I mean, I think it's more like seeing what works out for you. It's also, of course, there's kind of a catch-22. What we see with a lot of patients is if you're sick, it's hard to exercise. So that can become a problem. So we also might need some better ways to recreate some of the, some of the way that exercise changes the microbiome, we may want to be able to see if we can create some of that artificially, therapeutically, for people who are sick and can't exercise, so that we can get out of that sort of catch-22 situation and better allow them to begin to be able to move more, because it's difficult with patients who are ill when they, have exercise problems.
Isn't there some? Oh, sorry. Go ahead, Cosmo.
Didn't you just tweet yesterday, Amy, that there's recent data showing that when chronic fatigue patients exercise, the amount of lipopolysaccharide in the blood goes way up?
Exactly.
I was just going to say that.
That's one study, but yes, it did. This is a specific illness, but in these patients, yes, it did show that they exercise, but the disease is characterized by exercise intolerance, right? Patients can do things in the moment, but they cannot recover from that exercise. They get what's called post-exertional malaise, and malaise is a almost nice word to describe it. Basically just become derailed and they cannot recover. And so yes, in this condition, this one team showed that exercise actually seemed to promote bacteria moving from the gut into the blood and essentially increasing inflammation. So at that point, that's where personalized medicine comes in. You see, you don't want to tell that person to exercise to improve their gut health. You want to tell them to do something different, right? We need to intervene differently. So that's where these recommendations like exercise have to be taken patient by patient, because in some people, depending on the nature of what they're dealing with, exercise could be a problem too. So yeah, again, each patient is its own case.
I wonder if you could make the exercise prescription in chronic fatigue patients, you could tailor it. Maybe there's a certain intensity that if they exercise above that intensity, the gut is more leaky, or, So what's interesting is that, there's a scientist at Stanford, his name is Michael Snyder, and he's a huge quantified self type. And he wore a wearable, a fitness tracker for a long time. And he noticed that I think he sequences his gut microbiome, and not just bacteria, but viruses too. And that coincident with viral infections, he saw his resting heart rate spike. So there's something about, in exercise, you can over-exercise and you'll get the next day heart rate spike. So I wonder if those two are linked and connected. But that goes back to an intensity effect.
So if you catch the influenza virus, your likelihood of having a heart attack goes up by a factor of 14-fold in the 2 weeks following the infection.
Yeah, right. That's a thing. And then of course, if you get it, a lot of times you're treated with a whole bunch of antibiotics for whatever reason, because that's what we do, and a bunch of steroids to kind of knock down the inflammation, which are, can be profoundly immunosuppressive. So not only do we have the infection, but we treat it in a way that can diminish the immune response and the rest of the microbiome, which would allow the infection to have an even more permanently bad effect on the system. So we need to think about the way that we treat patients with these. Number one being, we need to stop prescribing antibiotics for viral infections, which we constantly do. It's out of control. People go to the doctor, they cough a little bit, and they're given antibiotics. I think antibiotics are very valuable tools. We need antibiotics. We just need to use them very carefully for very specific reasons when they are needed. And of course, that goes back to testing. We need much better testing so we can say, you're sick with X virus that works with this drug, or you have this bacterial organism. That means you should take an antibiotic. Right now, we don't do any of that. We don't test for any organisms. We just treat based off symptoms. We're just guessing. And that can be a big problem.
Yeah, definitely we need an improvement in the way diagnostics are covered out in medicine, especially diagnostics that we can follow temporally across time, which, that's another huge topic in of itself. But going back to the role of germs in the gut and kind of a little focusing more on the experimental aspect, I have a question here from Sven, which he asked a question a while back here. He wants to play devil's advocate here, and he's pulling up an observation that, I guess there could be a number of different ways to address this question, but according to him, germ-free mice do not live so much longer. I don't know exactly what that means, Sven. So much longer than their germ-exposed counterparts. How can this observation be explained? So Sven, does that mean that they only live a little bit longer, or they don't live longer at all? Something like 10%. Okay. So a very, very small I'm assuming, though, significant, right? So it is longer. But germ-free mice do not live so much longer than their germ-exposed counterparts. How can this observation be explained?
Well, you may think 10% is not a big deal, but 10% to a mouse, which is probably 4 months of an increased lifespan, 10% to a human, if you're going to go by maximal lifespan, that's 12 years. And if you're going to go by average lifespan, that's 7.5 years. I mean, this idea of live long enough to live forever, if you get an extra 10% by optimizing your microbiome or more, I mean, who wouldn't take that? So,
right.
Not to mention that their health is higher.
Yeah, better health span too.
Towards the end of their lifespan. And then we got to get into the question of, well, what is killing them 10% later? I mean, is it boredom-induced suicide?
I agree. And the conditions, I think we have to be careful to not overly extrapolate the mice. It's really interesting findings, but caged mice are not, they're not exercising, like Mike said. I mean, there's all these variables that are also affecting that. What are they eating? Mouse chow? Not sure what's in that. So, we're not even optimizing all these other things. Mice can have, who knows what's up with their immune systems a lot of the time. There's just a couple of studies which show that different types of mice react differently to different things. So mice is a great starting point, but I wouldn't over extrapolate like a 10% number as if it's going to hold up in humans. Although that it was a good question, but yeah.
So moving from the wet bench biology to computational tools. So this is a question for Dr. Michael Lascartes.
Yeah.
I've noticed in this seminar that you desire more computational tools or computational modeling to use in your research. What specifically would you like to see?
Well, I mean, tools, I can't tell you specifically because I'm not a computational scientist, even though I wish I were. But right now, the tools are very, they're, how can I say? So, you can do it the rudimentary method and look for associations between individual bacterial, at various levels, genus, phyla, class, or whatever, go down the list. And then there are better computational tools that can integrate, you're adjusting for age and BMI. But, I want, basically like MRI-based imaging and, DNA and RNA-based technologies to identify everything that's in the gut, not just one component of the bacteria. And the computational tools, being able to put them in a 3D map, how they interrelate. And it may exist. There may be 1 or 2 labs that are able to do this, but I mean, it needs to be ubiquitous. It needs to be everywhere so that basic scientists like me who don't have that experience can just use it freely without having to know how to code. So that's what I'd like to see. I can't give more specifics than that because I don't have a computational background. So
my background is pretty computational. The second chapter in my PhD was simulating a little mitochondrial channel protein in a supercomputer, and it took like 8 months to simulate it, and we simulated it for a total of 10 nanoseconds of movement, right? So at the atomic level, I'm not so excited about computational biology, unless you're talking drug binding studies. That's pretty fun. But I think the most excitement these days is in GWAS studies, like the UK Biobank, for example, finding all of these genotypes associated with all these different phenotypes as well. And what you said about doing MRI imaging, I think as we're moving forward, the new thing is going to be image-derived phenotypes, where we use the genome to predict features that show up in radiology scans. And there's a couple of groups doing that, really focusing.
Good stuff.
We have another question coming in from Elena. What symptoms can indicate that there are problems with the microbiome and it has to be tested and corrected? Like what signs to look for to suspect that there is something wrong with the gut bacteria? I just read that without pre-reading it, so it's like while I was reading it, I was thinking to the anchorman at the end. I was hoping that there was nothing, no word that was going to pop up. But anyway,
I would say diet-induced fatigue
or pain too.
Mm-hmm.
If you've got pain, it can't be a good thing.
And I'm not really going off of literature, just going off my gut in this situation.
Mm-hmm.
To be fair, most conditions are now tied to dysbiosis. Most inflammatory conditions are tied to some form of gut dysbiosis at the very least, if not dysbiosis of other microbiome communities. So quite literally, if you begin to have symptoms that bother you, especially GI symptoms, but there's a whole gut-brain axis. We didn't really bring that up very much either, where microbes in the gut communicate directly with the brain via especially the vagus nerve. It's a very important nerve that runs between the brain and the gut. And so what happens in the gut, for example, organisms in the gut can actually cause microglia to activate in the brain, right, via this nerve communication. So you could even have an increased neurological symptoms like anxiety or depression that are increasingly tied to dysregulation of the gut-brain axis and its ability to signal correctly. So there's a whole area of research also called psychobiotics, which are probiotics that people try to take to influence how the gut will signal to the brain to possibly control symptoms associated with neurological conditions. So really, almost any kind of symptom might, make you wonder at least what your microbiome is up to. As very broad range.
You just totally inspired me, and we need to connect on that. That's really interesting. I didn't know about the vagus nerve connection to the gliosis in the brain. I do know that obesity is largely associated with hypothalamic gliosis. That's the consistent finding. Gliosis in the hypothalamus. And it's those neurons that sense insulin and leptin and communicate via efferents like the vagus.
Right.
So I'm wondering if it's not the signaling per se, but it's microorganisms traveling through the nerve.
There's a team at Harvard and Robert Moore who works on that team. He thinks that organisms and some of the metabolites they produce can actually travel up and down the nerve. And there's a decent number of people who actually think the vagus nerve could be infected as well itself, which would also dysregulate signaling. So you have a lot of possibilities there.
Like in enteroviruses.
Enteroviruses being, or again, the herpes viruses, neurotropic viruses, and there's increasing number. So all of that possible too.
Oh, we gotta compare notes. I haven't blogged this yet, but the top obesity gene is actually a gene that binds to herpes virus in the nucleus.
All right. We got a message here from Alon from Scripps Research Bioinformatics Core Director. Sorry, I don't have a microphone or webcam, but I would be happy to discuss possible collaboration, grants, opportunities to develop tools to support microbiome and aging research. There you go. Email at adomisi@scripps.edu.
Are you working under Dr. Eric Topol, Adomisi? We'll see if you respond.
Not directly. Okay. Okay. All right. So we've got a couple of questions here. So one, then we'll backtrack to a stool question. Cosmo, could you please briefly explain what is diet fatigue?
It's just a personal observation that there are times when I haven't been taking my fiber, and it's probably TMI, but I almost died a couple of years ago due to a massive intestinal bleed. So I know that my gut barrier is terrible. Thanks, aspirin. But over the last several years, there are certain times where I'll eat something that causes joint pain, and I'll fall asleep for a couple of hours. I get so tired.
And just a question for me before we go into stool question. What in particular in your diet do you think triggered it? Is there, because we know that, for example, for joint pains, right, if you have high nucleic acid content for, like in shellfish, for example, you could lead to gout, for example, and that's well known. So in your case, were you able to pinpoint what was causing?
I'm not certain. I do know that if I really keep my fiber up, specifically if I take laxatives, that the problem goes away. A lot of the current work I'm doing right now is taking apart hematology analyzers so I could literally measure my immune system every day and see if I can correlate that with food intake. I built a food logging app a couple years ago that does barcode scanning and food recognition via photographs, so I'm probably going to revive that project. I want to figure out exactly what the molecule is that's doing it.
Okay. So Could we, okay, so one question that's popped up a couple of times here, is there anything about how our stools should look? Which I guess, I guess from a basic perspective, right? I mean, if your stools need to be brown because of bile salts, and that's indicative of that functionality of liver function. So that's something that's, if your stool is completely bleached, that's a bad sign. But other than that, to that extreme, is there anything else we should look out for?
You want happy stools, just like the poop emoji.
Happy
So there's
stools. It sounds like Bob Ross. Let's get some happy stools. Happy poop.
Well, if it was a poop emoji, it would be indicative of a stool teratoma, wouldn't it? But anyway,
there's something called the Bristol stool chart.
Yeah, right.
I've seen that. Yeah, I've seen that. And now I can't get Bob Ross out of my mind talking
that's
about.
That's the first time I've heard of that. But what is
one of my friends here in San Francisco made an app a couple of years called Baby Poo, and it's an app for tracking your Bristol score over time.
So just so you know, folks, glowing green, luminous green, not necessarily good. You might want to keep away from nuclear waste. Just saying. Or unless if you actually self-experiment and work at GenSpace, which is an organization I co-founded a while back, then it's probably a good sign. It means your project is working. GFP transplant. But anyway, so just a summary: the Bristol stool scale. What does that tell you?
I don't know a whole lot about it, but that's definitely where I would start if I felt like deep diving into poo.
And not all of us really want to deep dive into poo, but following on the stool question, can we talk a little bit more about fecal transplants? I don't have a specific question directed on this, but I guess maybe open the floor to, I guess, there's been a lot of papers on fecal transplants and purported benefits. So I guess maybe talk a little bit about that, because there's people obviously self-experimenting. There's people who have various gastrointestinal problems that seem to have been cured through the use of fecal transplants. So maybe, since this is something that people have been reading a lot about for the past couple of years, maybe let's talk a little bit about that.
So I know that it's commonly used for C. difficile infections. So the person that would have that, I think they would have to take antibiotics, and then they get a fecal transplant from a healthy donor who does not have C. difficile. So, but anecdotally, I know of one person, I think her name is Laura Peterson. I think she's a postdoc or scientist at Yale who had some prominent GI issues, and then she did a fecal transplant. And it cured, I don't know, at least she said she cured her whatever GI issue she had. So I don't think there's much data on, published data on healthy donors or generally healthy people who have some undiagnosed GI issue who then go and get a fecal transplant. It's mostly, as I said, in the C. difficile space. But I know that, I think George Church over at Harvard, I think he's been looking into athlete stool, not necessarily for fecal transplants, but just to figure out if there's a link between, a probiotic link to athletic performance. But I think fecal transplants are a little bit barbaric unless you would have to take it in the case of a C. diff infection. There's got to be a better way to improve the microbiome than shoving someone's poo where.
Well, what about certain diseases that are pretty extreme and the treatments are pretty extreme, such as Crohn's disease? What do you know about that? Is there any data regarding fecal transplants or any other type of gut inflammatory, diseases that where the treatment itself is removal of the gut, right? I'd probably want to try fecal transplant before I get that. Is there data that suggests that these fecal transplants are a benefit in those cases?
One of the problems we have is that the FDA has not necessarily decided how they want to regulate fecal transplants. Sometimes they want to call them a new drug. In that case, they would have to be tested in clinical trials that are very difficult to do for fecal transplants based off the individual nature of everyone's microbiome and the difference in donors and all that. So a lot of the fecal transplant stuff is happening at home right now, where people are doing it to themselves, I have to say. And so there's certain websites, certain forums where patients are basically just looking and they're trying to find the healthiest person they can find in their community or whoever they know, and they're doing transplants to themselves. I think what that number one says is the FDA needs to think about how to move this forward in a way that can be better regulated and acknowledged, because we're losing a lot of data by the fact that people are doing this in their own homes. So I can't really tell you very much about that, because I see patient cases all the time where people say, yes, I cured my Crohn's through a fecal transplant, but we don't have the data on, who the donor was. We don't have the data on how the microbiome might have changed over time. We don't have that. We're losing all that information because we're making it hard to do them in a realm that can be more regulated and studied. So I really think that we need to address that issue so that we can take a lot of this almost underground stuff that's happening with fecal transplants and bring that into the mainstream, so that we can better understand what's happening. One thing I'll say is that when people, if you're doing the fecal transplants on your own, think about, there's a lot you have to think about with a donor. Like I was saying before, the gut microbiome can affect also mental health in simple terms and neurological symptoms. So you want to think, people are using a donor, you have to think a lot about the donor's neurological health in addition to just their GI health. There are a lot of issues that can affect the microbiome. So finding a good donor is very important. And there are actually, I think, 1 or 2 companies that select donors. I guess there's one company that brags a lot that, not brags, that's just, I guess it's harder to be a donor for that company than it is to get into Harvard, because they're trying to find people who are incredibly healthy, have no symptoms. Because again, like I said, the gut microbiome can affect so many different symptoms in the body that you have to be careful in terms of who you use as a donor.
Right. So so yeah, it is kind of complicated to set up trials when you have such a, I guess, mix of microbes. But one very specific example that I'm reminded of in the literature is, some of you have probably heard of this, is the and this goes to probiotics, but it was isolated from a fecal sample from a soldier in World War I, which is the Nissl strain of E. coli, which is a strain of E. coli that was basically seen to be antagonistic to the growth of various pathogens such as I want to say Shigella, but basically, it seemed to inhibit the growth of intestinal pathogens. And I think it's approved as a probiotic. It's a purified, E. coli strain that's been around since 1917. This isolate, I think it's under the trademark of Mutaflor, and it's approved by in Australia and a number of other governments. I mean, you can buy it as a freeze-dried capsule, and so there's precedent for getting microbes from human fecal samples and then using them as a probiotic. Not a fecal transplant per se, but basically a fecal transplant, because these tablets are enterically coated, so they're protected as they go through the gut and end up finally depositing their payload of E. coli in the gut. So I'm assuming that there is a framework where such studies could be put together, where beneficial microbes could be isolated and then tested on individuals who have various chronic or acute conditions that are intestinal in nature. But again, I'm not an expert in FDA, mediated clinical trials. So.
I mean, going back to the computer modeling based approach, right? So if you knew that someone's got some GI condition and you've got a certain probiotic or certain strain of whatever bacteria, bacteriophage, fungi, whatever, ideally I'd want, before that was even taken as a supplement, I'd want to see computer modeling of that person's gut, and then computer modeling of after you've taken the probiotic, how it changes the gut. But not just that, you've got to also take into account the person's own DNA and their diet and their exercise and all these other environmental factors, how it would potentially interact with a potentially new microbiome and what would be the long-term effects other than potentially changing one part of the microbiome. We're nowhere near that day now. I mean, it's almost like the antibiotic approach, nuclear bomb the whole facility just to kill one bugger, you know what I mean? And then hope you get, a healthy microbiome that grows out of that. But I'd like to see more advanced, computer computational-based tools that can address these things first, rather than the trial and error approach, which will take 50, 100 years, who knows.
All right. Do we have any more questions coming in, Steve?
I don't know. Let's ask our moderators.
Elena asking about your take on antibiotics, gut health, and antibiotic resistance.
I mean, we kind of touched on it earlier that, you know, ideally there are obvious situations where you would need an antibiotic for a certain pathogen. You know, if you're just going to be prescribed antibiotics because you have a cough, Amy was talking about that, it's ridiculous, right? So, and, you know, it's not just people taking antibiotics. If you're a farmer and you're not feeding cows grass because that's what they evolved to eat, and you're feeding them corn, and now they've got massive infections in their gut because they haven't evolved to eat corn, and then the farmers are going to go then and give them antibiotics. Well, now your meat, if you eat meat, has antibiotics in it, and I think there's some evidence that there are antibiotics in the water. I mean, it's everywhere now. So when you have antibiotics everywhere, you're going to end up with antibiotic-resistant bacteria. It's just there's no way around it. You obliterate the species, but the ones that evolve to be resistant to it will exist, and then you need new antibiotics. So let's hope that we can develop new antibiotics before we revert back to diseases that we've eradicated. You know, measles, mumps, rubella, all the things that we have nice vaccines for. So.
Okay.
Yeah, one thing I'll add on that, though, is that I think we need to keep the state of the immune response in mind, which is something that I mentioned earlier, in that a lot of these superbugs, if you want to call anti-resistant organisms, tend to be the worst in people who are immunocompromised. So especially in people who unfortunately have to take a lot of immunosuppressive drugs because they've had a transplant surgery, things like that, hospitalized patients. So what that suggests is that if we can improve, continue with also therapies that keep the immune response in better shape, we may be able to prevent more microbes from actually becoming resistant to antibiotics. You'll see a lot of cases are cases in which patients are taking immunosuppressive drugs are where you see a lot of the organisms turn into the most difficult resistant forms too. So we have to keep the immune response in mind.
All right. So, Steve, I think you sent another question here to the group. Thoughts on fasting in the microbiome, and should we? I think we should wrap up on this question since we've talked about diet. So this is a great bookend, which is, well, anti-diets, fasting and the microbiome. We know fasting is good. We know periodic fasting is you know, good. It promotes autophagy. It does a lot of great things to your body. Does it do any great things, and I'm assuming it does, to your microbiome? And what are thoughts on fasting and the microbiome and the connection between fasting and the microbiome? And are we still really, well, I think all of this research is still kind of early, but particularly on fasting and microbiome. Thoughts?
So I haven't looked much into it, but the microbiome, gene expression of the microbiome is there's actually circadian rhythm. So I wouldn't be surprised. But then it goes back to the individualized thing, right? Somebody may be able to eat all of their food or whatever at 10 o'clock at night and not affect their microbiome composition, and whereas others can eat it at different times. But I think, again, that's individualized. You'd have to figure out what works best and how does it feel best to eat at certain times or not to eat for certain times, as in with fasting. There may be a paper or two on fasting in the microbiome, but it's not my field. I haven't studied it, but I'm pro fasting.
Okay. So I think some of the take-home issues here is that we do need much better diagnostics. You know, I'm hearing that if you want to measure your microbiome, a lot of this stuff is, you know, for better or worse DIY. I think the positive aspects to our situation right now is that we have the internet at our disposal, so we can look all this stuff up. The problem is that we have a torrent of information and misinformation that we have to wade through to get to the right, you know, to get to the right source. I think it's very useful knowing some of the companies that were mentioned, some of the sources that individuals can go to if they're interested in looking up their own gut microbiome health, or the, you know, the composition of the various genuses of bacteria that are in their gut. But a lot more needs to be done as far as knowing what else lives there, or, you know, live is sort of a loose term when it comes to viruses. But you know, medicine is still progressing, and it hasn't caught up with a lot of the science, unfortunately. So, you know, you can't go to your doctor's office that I'm aware of unless you have a very savvy doctor or some center that's really specialized in this and basically get your gut health microbiome readout and get some sort of, you know, predictions made out of it. You kind of have to do this yourself, is what I'm hearing. The literature is out there for you to look at, and, you know, and there's private and, you know, and privately funded and publicly funded sources out there where an individual who's motivated, and I'm sure we'll share the links here, can go and basically look this up on their own and take a more proactive approach, and certainly reach out to the people that have been on this panel.
Oliver,
yeah.
Real quick, just to add in just a little bit to what you said, not just the gut microbiome, but for example, if I go to my dentist and I say, this is what I'm doing for my oral health, like I make sodium bicarbonate xylitol solution as mouthwash to keep my mouth basic, because the oral pH should be around neutral. And anyway, so I expect the dentist should have some expertise on my oral microbiome, and I get crickets every time I go and every time I ask. Crickets. They have, unfortunately, or maybe it's just my dentist, I don't know, no idea. So yeah, again, like you said, it goes back to individual responsibility and as many people educating themselves in this as possible, and that's what we can do for now.
Well, you know, hopefully, and I think everybody here is on the same page, we're getting to a better world where all of this information, all of this expertise is not siloed, so to speak. It's more holistic. You know, just the fact that you mentioned, you know, you went to a dentist, and our oral health is completely disconnected from the rest of our body. It's in our head, for God's sakes. It's right underneath our brains, right? And somehow it's seen as a part of the body that's floating out there, disconnected from the, you know, it's very arbitrary and medieval designations. Sort of reminds me of the 4 or 5 humors, right? I specialize in yellow bile. This guy specializes in black bile, and that, and you know, and it's all integrated, of course, and it's sort of arbitrarily treated as distinct and separate, as if you know, a tooth abscess can't kill you, right? Or when it does kill you, then people suddenly realize, oh wait, it's connected to the rest of your body. So you know, that is a general medical health issue. And when I say medical, I'm involving everything: mental, dental, you know, gut, everything. But that's a problem that we'll definitely have to address in another webinar. Maybe a number of webinars, not just one. But I thank everybody here for joining in on this discussion here. Certainly the microbiome, not just pertaining to the human health you know, environment, but also the global health environment. The microbiome goes all the way up to the stratosphere, on down to, we don't even know how deep the microbiome of the earth goes, right? As far down as we drill, we still start finding microbes. So microbes are permeating this entire, you know, this earth, and they're definitely playing a huge role in our varied ecosystems. And the human health microbiome is just one, albeit very important, aspect of the global microbiome. And just for our, you know, overall health microbiome, we are still kind of taking baby steps towards finding out how it impacts our health. And, you know, all the studies that are coming out strongly suggest that it's having huge impacts on our health. So I thank everybody here for contributing to this discussion and basically giving a lot of your insights, and also some really helpful practical advice as well for people who, you know, want to go that direction. And thanks to everybody at LEAF, Life Extension Advocacy Foundation, for making this all possible. People that are helping out in the background here. Nicola, Victor, Sven, Steve, Fatima, who's somewhere in the background, flipping switches, and our panelists, Amy, Cosmo, Mike, all doctors, and all experts in their respective fields. So thank you, everybody.
Take care.
Thank
See you later.
Thank you to the heroes as well. We'll see you next time. And
of course, the heroes.
Of course, come on. I mean, you know, they're the reason we do what we do. So thanks, everybody, from my point of view, for coming. It was great. I mean, we could do a dozen shows. It's fascinating. Things I learnt today. It's just unbelievable. So fantastic. So thanks everybody. And maybe we'll do a not microbiome. Maybe we'll do a biomarker one next time because I know Mike's itching to do one of those. So, let's talk buns and CRP and.
All right.
Yeah, he's always talking about his buns. That's one thing you get.
Blood, urea, nitrogen. Blood, urea, nitrogen. Yes.
Oh yes. Yeah. Well, you didn't think I was talking about your other buns, right?
Ah, funny guy, Steve. Funny guy.
That's what BUN stands for.
Oh yeah.
Okay, with that, now we're getting into. All right, so are we officially done?
We
All
are.
right.
So
Take care, everybody.
Never.
we're never done.
The joke never ends. Okay, take care. Thanks, everybody.
Thanks, guys. Bye.